Welcome. Thank you, everybody. We are presenting today on open data and tools in education, which is a panel presentation that we presented at the CellBio 2022 conference that we're excited to share with you today as a webinar. So on today's agenda, I'm going to introduce the Allen Institute's Open Data and Education program. Then Gideon Dunster will introduce the Allen Institute for Cell Science, one division of the Allen Institute that the rest of our speakers will be concentrating on today in their sections. Tom Martinez will speak about reaching high schoolers with open science. And then Sarah Latchney on case studies of teaching college neuroscience and cell biology with open data. And then to conclude, Carlos Goller will present a lesson that he developed with us here at the Allen Institute called Does Organelle Shape Matter? So to start, about the Allen Institute, our open science and our education program. The Allen Institute is a biological sciences nonprofit located in Seattle, Washington, with focused research areas in neuroscience, cell biology, and immunology. The Institute also supports cutting-edge research across the world with the Paul G. Allen Frontiers Group. Across the institutes, our scientific process is focused on tackling complex, broad, and hard problems in the fundamental sciences. And our approach is guided by three core principles: big team and open science, which I'll return to in a moment. And our work generates data, knowledge, and analysis tools, all of which we share openly and publicly. Our core principles drive the work that we do and how we do it. We work on large interdisciplinary teams that tackle complex challenges requiring many different areas of expertise. As of fall 2022, we have about 140 scientists with doctorates in over 30 different fields. Collectively, these teams generate very large data sets. And we share that data and analysis tools, our findings, and research tools like cell lines publicly for anyone to use. The majority of our users are using open resources to support their research, but we also have our education program to help teachers use our resources to provide research experiences to students, primarily at the high school and college level. So the Allen Institute Education Program, our resources are available at alleninstitute.org/learn. We offer programs like field trips, classroom visits, both live for those in the Seattle area and virtual for those anywhere else, webinars for students and educators like this one, and new in 2023, we're adding a teacher academy. So stay tuned on our website for upcoming sessions. We offer resources like curriculum units and lesson plans, some of which are virtual cures, which I'll return to in a second, and other classroom activities like brain bingo, everybody loves bingo, as well as programs that give insights into different career tracks in science, including some that are easily overlooked, like software engineering and project management. And our unique value as a program is using open science for education, giving students access to research grade tools and data that they can use in their classroom right there in the browser window. So what can students do with open data? Students can grow along this axis of sophistication and independence using open data to support their learning in different ways and at different levels, from exploration to guided research to fully independent research. The Allen Institute's open data encompasses a wide range of data types, from in situ hybridization to single-cell RNA sequencing, multiple types of electrophysiology, two-photon imaging, and data sets that combine multiple data types, covering neuroanatomy, cell biology, and immunology. The Allen Institute doesn't release the data only when we're done analyzing it. The data sets are too big to ever really be done analyzing them anyway, but we release them as soon as they're quality controlled and ready to be used by outside users. There's lots of opportunities for students to have genuinely novel discoveries when working with Institute data, let alone the expanded opportunities that they can have when they combine their own data with Institute data. On the less intensive end, students can simply explore some open data on our websites, see what it entails, learn about the methods, and poke around some images and visualizations. We're actually interested in the impact of just mere exposure to open data on student learning outcomes and enthusiasm for science, as well as what happens when they're engaged at a more intensive level. The next level of involvement would be to do a lab module. And because everything is online, you don't have to have access to a lab to do this. So it opens up opportunities to participate in research at this small scale, all the way through independent research projects to students and institutions without lab access, and to types of data that are not typically accessible to undergraduate labs, like human data, or techniques not typically feasible in a teaching setting, like electron microscopy. If you do collect data in your class, students could collect data and then use open data as a comparison, a control condition, or even the main focus of their analysis. So they get both exposure to a lab technique and analysis without having to worry about whether their data came out well or whether they have enough of it to get really good statistics. I'm going to show you a few lab modules that we have available that use our data for a fully online experiment in just a moment. The next level of advancement and independence is to do a full CURE, course-based undergraduate research experience, where students don't just do an experiment that we've laid out for them, they design their own. This could be fully using open data or in combination with student-collected data. And finally, there's the whole world of independent research outside of a class context, which could be in conjunction with students' own data or not, especially for very computationally oriented projects. And this can be a student's entire thesis project for an undergraduate or even their entire dissertation for a graduate student. All of these different types of learning experience can be executed in settings that are fully in person, hybrid, or fully online, giving remote learners a form of a lab-based activity that they might not otherwise get. And the level of coding capability required also ranges from none to expert, with opportunities for students to grow their coding skills as well. So I have here some samples of some of our education resources that use open data. We also have programs for educators using our open resources to help inspire the next generation of professional scientists by giving them these experimental experiences. We also offer webinars for educators demonstrating how to use many of our curriculum resources. Again, you can learn more about our education lesson plans, our webinars, and all of our other programs at alleninstitute.org/learn. The Allen Institute Education Program covers multiple divisions of the Allen Institute and research areas, all of which use our open science to support these research experiences. So today we're primarily going to be talking about some specific case studies using data from the Allen Institute for Cell Science, but also from the Allen Institute for Brain Science, which is our largest and oldest division. So the Allen Institute for Brain Science is interested in developing a parts list of the brain across species, across developmental time, across brain regions, and across different modalities. This parts list means assembling a catalog of all of the different types of brain cell that are found in the brain in these different conditions. And that includes both neurons and non-neuronal cells. They're mainly looking in healthy brains as well as in some selected diseases across these multiple modalities. And then the Allen Institute for Immunology is interested in developing a profile of the human immune system across health and disease. So they're looking at healthy individuals and how their immune systems respond to a challenge, such as a flu shot, as well as to a hyperimmune state. The immune system is responding to things that it should not be responding to, in particular, looking at irritable bowel and rheumatoid arthritis, and also hypoimmune states when the immune system is not responding to things that it should in cancer conditions that affect the immune system, leukemia and lymphoma. They just released their first data sets in 2022. So please go check those out as well. We'll be talking about some of the brain science and cell science ones today. So with that, I'm going to hand it over to Gideon Dunster, who is going to talk about the Allen Institute for Cell Science, which is the source of a lot of the open data that our educators are going to speak on next.
Thank you, Kaitlyn, for that wonderful introduction. And I'm excited to talk to you all about the Allen Institute for Cell Science. My name is Gideon Dunster, and I am an Alliance and Project Manager here at the Institute. So let's dive right in. So like all of the institutes, all the scientific divisions that work under the umbrella of the Allen Institute, we have a pretty large scientific goal that we set out as our main purpose. And specifically, our goal is to understand the organization and behavior of cells as they go through their life cycle of dividing, living, and eventually perhaps even death. And in some cases, we delve a little bit into differentiation. And we do this using human-induced pluripotent stem cells. So these are stem cells that came from an original host that were used to induce pluripotency, so back to stem cell state. And most of our research focuses on cells that grow in colonies in that state. And our idea is that if we can build a map or an understanding of the cells as a whole, we can use those as reference points for other researchers that are interested in applying and comparing, perhaps a healthy state or an undifferentiated state to cells that are diseased or as they go through differentiation. So another way to think about this is we use some of these allegories. One is the periodic table of elements, or for those of you who have an evolutionary background, the theory of evolution and Darwin's finches. And in both of these cases, right, we're talking about heuristics, looking at relationships in a system, but taking a broader view than looking at any single isolated point. So for example, while previous or current researchers might work on a single element within the periodic table, or biologists might really be interested in understanding the ecological niche and behavior of a single finch, we're more interested in taking a step back and looking at broad views of how these elements or finches might build together to build a coherent view of how cells are going to function over their life cycle. So we're looking at trying to to create heuristics that look at this system as a whole to be a reference for other researchers. Now, how would we want to do this? Cells have lots of activity, and there are many different scales at which we could approach this problem. So for example, we could look at all of the genes and transcripts or the proteins that happen within a cell's life cycle and try to track all of their expression and activities. But as you can see here, that scale is so large that it's almost too big for even our team of 70 scientists or so to try and approach. And so instead of trying to get down to that granular level, we decided to focus on organelles, the major subcellular structures that are responsible for most cell behavior. And we did this because while it is a large problem, tracking and understanding the activity, location, and behavior of 30 to 50 subcellular structures over time is a big problem, we still thought and still maintain that as a manageable problem for our team. And so what we tried to do is we try to approach this kind of using a holistic team science approach. So whereas academic labs have individual PIs that focus on singular research questions or maybe multiple research questions that are related to one another, and those PIs operate kind of separately from one another, we focus on a team-based approach where we have one large overarching scientific goal, and then we have teams along the way that all contribute to that major goal. And I'll walk you through that here in just a second. So how do we do this? Well, there are three main steps to completing this research. The first is, can we identify and target a organelle of interest so that we'll be able to make it glow? In this case, can we see it? So we try to target proteins that are tightly associated with given organelles and see if we can't insert some sort of fluorescent protein to make them glow. Once we have those, the next question is, can we see them? Can we take images of those organelles as the cells are living in their colonies at a resolution and a time scale that allows us to make sense of those behaviors? And lastly, can we then take those images and can we analyze them to pull out data that will allow us to say something meaningful about what we've observed in those colonies? And so I'll break these down one by one and show you a little bit about how we do it, but also what kinds of things that we then release as an open data research institute along the way. So the first step is, can we make these key structures in the cell visible? And as I mentioned, we use CRISPR-Cas9 to try and insert a fluorescent protein into a protein that's tightly associated with an organelle. Frequently, this is GFP, but sometimes we may choose different fluorescent proteins. And in general, we tend to target one organelle and one protein at a time in each of our cell lines. And we go through extensive quality control testing to make sure that by the insertion of these fluorescent proteins, we aren't interrupting any of the normal cell behaviors because we want to get a naturalistic view of the cell behavior as possible. And over time, it turns out that we've gotten actually quite good at this. This is a subsample of the 54 cell lines that we currently have released through Coriell. So if you're interested, you can always go to their website and see about picking up a vial of cells for yourself, which you can then use to maintain your own colony. And in each of these pictures, you can see the major organelle or structure that we have tagged, and then in parentheses, the specific protein that we inserted our fluorescent protein into. So you can see we have things like the mitochondria, endoplasmic reticulum, and Golgi, but also some things that like key structures like actin filaments. Excellent. So we have now shown that we can tag these key structures. The next question is, can we see them? And can we see them at a resolution and at a time scale that allows us to make sense of the data? And so in this case, we use a variety of different high-powered microscopes at the Institute, kind of like having a tool chest. Each microscope is used depending on the specific question that we have. And we end up taking lots and lots and lots of pictures. We do this using live 3D cell imaging. And so you can see we have a variety of different microscopes within the lab. And these are tools that are constantly evolving and changing based on our research question and the needs that we might have. Great. The last question is, okay, we have generated hundreds of thousands of images. Can we make sense of them? Can we actually pull out the organelle of interest? Can we track it over time? Can we compare it to cells of different organelles that are tagged? And can we say something meaningful about those images, right? Because just having pictures doesn't really tell us anything statistically about the structure or the activity of the cell itself. And so to do this, we end up using a good number of quantitative approaches, of which we have several teams that focus on different aspects of this. And one example is shown here is the ability to look at the structural variants of a cell. So these are some shape spaces that our teams have identified that occur naturally in cell colonies. And you can see that we've tried to standardize the definition of these shape spaces and look at how they can vary across different cells over time. And along the way, we create opportunities to share all of this information and these data with the public. So for example, all of those images that we take of the live 3D cell images, those are published online, both in collections that can be used by educators, but also in collections that can be used by researchers that you can run your own analyses on them. The tools and resources, the software tools that we use to extract, for example, certain structures and make sense of the 3D shapes, those software are also published online in open source spaces, and more often than not run on Python, an open sourced coding software platform that allows researchers, educators, and students to use those tools and even repurpose them for their own research. And so here's some examples of the ways that we have applied those data to lesson plans and with a focus on education. And so some of our other educators on this panel will talk to you in depth about the ways that they have applied them in their own classrooms. But if you're interested, you can always learn more on our website to see if there's something else that might fit your needs. And with that, I will turn it over to our first panelist, Tom Martinez, who will talk to you about using some of our resources in a high school setting.
Hi, my name is Tom Martinez. I'm a retired science teacher, primarily biology and all forms of biology education, from the most basic forms of biology, dealing with freshmen in high school, all the way through AP biology and biotechnology. My career has primarily been dealing with students in a public school setting, 39 years worth of public school settings and 28 years worth of public school education in the western suburbs of Chicago. My students are very diverse learners, some extremely high performing students and some not so highly performing students. But the point here is that I want to try to make sure that I am giving my students the opportunity to understand biology a lot better than the way in which it was done in the past. When I first heard about the Allen Institute and open data, I was at a stem cell symposium at the University of Wisconsin, and the speaker had mentioned the fact that the imagery from the Allen Institute could be used as educational material that could further enhance the ability to understand what cells are and what cells do. And so in the high school classroom, looking for outside resources is really important because it's primarily book-based. And I believe, as many teachers do, that there are a lot of misconceptions and difficulties with conceptualization using just book-based resources. And so using the outside resources has enabled me to take a deeper dive into what cells actually look like and actually what they do. And I can tailor that imagery to the level of my students based on whether or not they've had previous experience or not to cell education. And so for me, and I think for many teachers, there's a lot of different kind of interactions that can be done to make learning successful. And I find that tactile and visual and kinesthetic ways of learning are the best. And so the open data and the imagery provided by the Allen Institute allows me to tackle that idea of visual. And many students are visual learners. And so I think this kind of fits that niche. And so this is an image of your typical cell found in the textbook. Still being used in textbooks today, and it's great if you want to identify structure by shape, but it has nothing to do with location within the cell or actual appearance as the imagery would suggest it does. And so this is a nice little introductory image, but I wanted to go deeper. I wanted to go farther into what cells actually look like and how they perform. And so when we look at the imagery available from the Allen Institute, we find that the images look completely different. And they help to eliminate any of the misconceptions that are derived from textbook imagery. And so what we can do is we can take a look at cells and what they really look like at any particular time, including subcellular structures. Where do they go? How do they behave? If a cell is dividing, what happens to those structures? And this can be identified and tagged through the imagery. So the library has over 300,000 images that are available. And the ones you can see here on the screen, when I first saw them, blew me away because this doesn't look like anything that you would find in a textbook. And these are actual images. And I think students prefer that. They want to know, is this actual or is this something that has been created by an artist? And so, for example, one of the difficult things that my students had trouble with was understanding what happens during mitosis and this concept of microtubules and controlling chromosomes. And again, you can see this on the imagery as it actually occurs, so that we get at least kind of a reference point so that we can go deeper into how this occurs. I can add and subtract cell structures. So if I want to, say, for example, working with some of my more advanced students, if I want them to tag a particular structure, research a particular structure, they can do that. And I have a list of items that can be identified at any given time in the life of a cell for a student to take a deeper dive with. I can also look at more fundamental cell structures using some of the imagery, not only for cell functions, but also cell structures. And I would use this maybe for my more introductory students who are first time looking at what goes on within a eukaryotic cell during any point in its life cycle. I can then use some of the information derived from the site to identify particulars. So for example, if I want to look for how the mitochondria is behaving or where the mitochondria go, I can tag that. I can also select from what phase of mitotic division the mitochondria are acting or where they are particularly going at that time. I can look at the tutorials if I want to identify mitochondrion, for example, and Tom 20, I can tag that and find out what's going on. I get a tutorial on the right-hand side to direct student understandings. So it's a beautiful way to identify not only structure, but function, and also get a tutorial along the way. If I want to take a little deeper dive, I can do that. Again, this is based on student level and familiarity. And so by having access to data that's provided by the Allen Institute, I can shape, I can construct a learning environment that is appropriate to my student understanding. We're also working on what happens when mitosis goes wrong. And so we're working on a piece that deals with mitosis in cancer using real imagery, real pathology imagery. We're looking at tumor imagery, we're looking at gene expression information, and also drug efficacy data to identify how do we treat a particular cancer like glioblastoma. And so when we look at a different look at mitosis, not only can we look at it from a basic standpoint, but we can also move to a more specific, more advanced standpoint, and what happens after mitosis or if mitosis goes wrong. We also look at quite a bit the structure and function of neurons. Most of my students have never seen a neuron. They may have heard of the word neuron, but they can't visualize it. And so the Institute has unbelievable imagery of neurons to enable my students to get an idea of what these things are. There are standalone curriculum that's been written, but again, you can take any of this data and plug and play depending upon the level of student understanding and interest. And so in terms of using the Allen Institute open data, the information that's given online, the information that's available online for teachers is vast and expanding. And what I find is that it frees me up as a teacher to use the resources as I need. I've rarely used a standalone curriculum start to finish. I'll plug and play. And it saves me time, but it also allows me to build a more comprehensive understanding of what cells do, what they look like. So where do the organelles go when structure and function is at play? And so in my experience, using this open data to enhance my teaching, it's been a great resource for me to bring my students to a level of understanding that I need them to be at. Thank you. And I'll pass this along to Sarah.
Hello, my name is Sarah Latchney. I am an assistant professor of biology and neuroscience at St. Mary College of Maryland. St. Mary College of Maryland is a small public undergraduate institution in Southern Maryland. I really got into using some of the open data set from the Allen Institute at the height of the COVID pandemic. In the fall of 2020, I was teaching a hybrid format where I had half of my students were remote at home, streaming on Zoom, and the other half of my students that were physically present in my classroom. So I had to develop some laboratory that would engage both types of learners, with the idea that I wanted to have both my in-person student and my remote student to have equal and equitable access to the laboratory. So to do that, I turned to some of the open data set that the Allen Institute has to offer. So one example was for my 100th level introductory biology class. We were going over mitosis. And one of the problems with teaching mitosis is that many high school students come in having already learned about mitosis in high school, but they really don't have a good understanding of the terminology and the different types of structure involved in cell division. And they're also very used to seeing very clean, rudimentary sketches of cells in different phases of the cell cycle and mitosis. And with this, it really lacks some real life authenticity that comes with learning about mitosis. And the students don't really develop the skill to analyze and quantify scientific data. So what I wanted to do was to work with my students to have equal access to the laboratory and have opportunity to develop scientific hypotheses based on what they already know about the cell cycle. So what I did was, there is a publication that was published by the Allen Institute. And they had the massive open data set available online where they have virtual confocal images of cells in different phases of cell division. And so what these students do is not only they have to describe the main features of different stages of cell division, but they have to use the dynamic open source data set to develop a hypothesis about how long the cell will stay in each phase of the cell cycle. But they have to do this by looking at real confocal images rather than clean textbook images. And what they do is they quantify the relationship between different stages of cell division and how often the cells are present in each stage of the cell cycle. And they use the data to either support or refute their hypothesis. This is an example of a screenshot of one of the open source images. And what they can do is they can manipulate these images and show, say, the genetic material or the cell membrane of these cells in different phases. And they have to identify if the cell is in interphase, prophase, metaphase, anaphase, telophase. And this really forces the student to be able to recognize different cell structures rather than just looking at a clean textbook image. And so by doing it, they really develop and solidify their understanding of what different chromatids look like and how these sister chromatids separate during mitosis. And you can see on the right-hand side of the slide, these are examples of the images that students can analyze. And they look very different, but also very authentic compared to the images that they are used to seeing in a textbook. So that's one example that I have used in my teaching. The second example that I've used for my teaching was for a 200-level introduction to neuroscience class. In the class, I also have a similar objective where I'm really trying to get my students to understand that not all neurons look the same. In fact, neurons can look very different. But the textbook image that you see here on the left, this is a very typical image that students are used to seeing, where they have the main cell body, a main axon, the conduct, and then you have the axon terminal. But I really want to get students to understand that not all neurons look like that. And also give them opportunity to develop hypotheses, be able to test their hypotheses using these open source data sets, and also develop their analytical and quantitative skills. So for this data set, again, the learning objective is pretty similar to the learning objective for the mitosis lab, where they are trying to compare the morphology of neurons that they're used to seeing in a textbook to neurons that have been reconstructed using the open source data set. And specifically, they really learned to understand to learn how axons look very different from dendrites. And they also compare the morphology between excitatory neuron and inhibitory neuron. And they start to understand that these morphology can be very different. So this is a screenshot for one of the other data sets, and the link is right here. And what you can do is you can classify neurons based on their electrophysiology. You can also classify them based on their morphology. And they have different filters available here if you want to look at how neurons might be different in human versus rodent. You can look at excitatory neuron versus inhibitory neuron. And what you get is the electrophysiological data up here that you can analyze. And you also get the morphological data that you can look here. And so again, students develop hypotheses about how different neurons might look different, or different neurons might have different electrophysiological characteristics, and they use a data set to test their hypothesis. And then the last case study that I have for this seminar is some of the other data sets that I have used for my upper level neurobiology class. For this course, I have developed a multi-part laboratory where I want students to first analyze how genes might contribute to aging and dementia. And what they do is they navigate yet another data set where the link is right here at the bottom. This is the Aging, Dementia, and Traumatic Brain Injury database. And what they do is they learn how to look up the function of different genes that they might be interested in. And they analyze the gene expression in the Aging, Dementia, and TBI database. And what they do is they learn how to download genetic information in the form of an Excel spreadsheet, and they use statistics to analyze the data. They develop hypotheses for how their gene of interest might be related to aging and dementia. And with these hypotheses, they develop their own independent project for exploration. And then the second part that I get to, they get that genetic information that they've been analyzing, and they link it to learning and memory behavior, and try to tie it all together and understand how genes and behavior might contribute to various neurological or psychiatric diseases. So this is the screenshot that you get from the Aging, Dementia, and TBI database. So what you can do is you can enter your gene of interest, and you can also analyze it by different areas of the brain. So in my case, I was interested in the gene P10, specifically P10 in the hippocampus. So P10 is a very important signaling molecule. It's a phosphatase that has been shown to be involved in some learning and memory. And you can see at the bottom, this database will generate a heat map. And you can look at all the different human donors and analyze if P10 is upregulated or downregulated based on whether or not they have dementia. Or you can even filter it by biological sex. You can filter it by age. You can filter it by just different characteristics of Alzheimer's disease. And so the students play around with this heat map, and they filter the database based on what their hypothesis is. And they download the genetic information, and they use that information to basically support or refute their hypothesis. And so then, like I said, the students take it one step further with the second part of this laboratory, where they will link whatever gene that genetic information that they have downloaded, and they link it to behavior. So to do the behavior aspect of this laboratory, I use this microscopic worm, basically, known as C. elegans. And the cool thing about C. elegans is that about 40% of the gene have homology to the gene expressing human. So the homologous gene for human P10 is DAF18 in C. elegans. And what you can do is you can purchase C. elegans that are wild type, or C. elegans that have a genetic mutation for DAF18. And you can investigate how DAF18 might be involved in learning and memory. So on the left here, this is the online database from the Allen Institute, where they looked at gene expression. And this is the C. elegans behavior kit that I had ordered from BioLab. It's a very easy step-by-step kit that you can use. All the instructions are provided by the company. And with these two parts, I linked them together with my student. and that formed the basis of the independent research project. And so now I'm going to hand it over to Goller for him to talk about what he had done.
Thank you, Sarah. My name is Carlos Goller, and I'm at North Carolina State University in the biotechnology program. And I had the privilege of working with Kaitlyn and several members of the Allen Institute to develop a case study that addresses high throughput imaging and brings those technologies and the analysis of those data sets to the classroom. And we created an activity that we called Does Organelle Shape Matter? So how do we bring HT or high throughput biotechnology approaches to undergraduate and graduate biology and molecular biology courses? High throughput technologies are becoming increasingly common, yet students often don't have the opportunity to work with some of the instruments or analyze some of the data produced from these approaches. Thus, they have a limited experience that is becoming more and more critical in the workforce. So what we did was we worked together to create a case study that brings open data sets and high content microscopy to undergraduate and graduate labs and courses. And to do this, we used a case study approach. We came up with a hook, a fictitious scenario. We really emphasized an important new cutting edge approach, and we used the open data sets from the Allen Institute. And together with Graham Johnson and Kaitlyn, we were able to create and publish a lesson in the journal Course Source, and that a screen capture of the lesson, as well as a link to download the documents, and then the lesson plans are on the slide. So what did we do? How does organelle shape matter? So our lesson had several goals. What we did was we really wanted students to understand how structure of organelles varies within cells and over time. And to do this, we focused on the endoplasmic reticulum. Our second goal was that students should learn about the real world applications and interpretations of high throughput microscopy, data sets, and the methodologies behind the creation of these really intriguing data sets. Finally, we wanted students to learn from open data sets and how open data sets can be references that provide context for their own data. To do this, our lesson really focused on learning objectives that increased and walked students through understanding key facts, all the way to evaluating hypotheses and creating their own experimental design. So our learning objectives were to describe the morphologies, that is the anatomical structure of the endoplasmic reticulum and its primary functions, describe the purpose and applications of high throughput microscopy, compare cell structures using general visual analysis tools, and in this case, we demonstrated using the 3D cell viewer on allencell.org. And then we challenged students to design a future experiment using a similar data set that builds on their findings. To do this, our setup and logistics relied on understanding our classroom context. So we had a lab-based course for undergraduate and graduate students, and we tailored our lessons so that they could be implemented in a variety of courses, including molecular biology, genetics, biotechnology courses at both the graduate and undergraduate level. We wanted students to be able to work individually and also make the case study adaptable for group work. The activity was also implemented before and during the pandemic. So we added adaptations and made modifications for it to be deployed synchronously as well as asynchronously. And we did this using both hard copies as worksheets and also digital versions that allowed students to submit responses and share their work digitally. The lesson plan consists of the following steps. We wanted to determine ways to distribute the handout to students, and this could be electronically or physical handouts. We then introduced the learning objectives and really connect this to why high throughput microscopy can be so useful. We introduced the scenario for the case study that I'll discuss in the next couple of slides. And then we determine groups if we do this in groups. We provide the guidelines and the time frame for work, usually one session for group work or for example, 1 week of work online asynchronously. And as submissions come in, we always try to do a debrief either in person in the next in-person session or online through discussion forums and maybe a short summary video. And a table with a example timeline for the activities is included in the course's publication and includes preparation before the class, the introduction, the case study itself, the reflection activity, and how we review work, as well as estimated times and some notes for implementation. So at NC State, we implemented this in a lab-based course that we call High Throughput Discovery, in which undergraduate and graduate students work together to learn about different high throughput sequencing, high throughput microscopy approaches. The parts of the lesson include a scenario in which a Dr. G, which may be Dr. Goller, was talking to his graduate students about an interesting effect they see of an antibiotic compound they are working on, and how this compound may be affecting the endoplasmic reticulum structure of the cells they are doing viability tests on, eukaryotic cells. Part 2 asks the question, what does an endoplasmic reticulum look like? And we compare the textbook version to the images that the Allen Institute has made available through this public data set. Part 3 focuses on different cells. Let's get quantitative. So we start to use volume measuring tools available on this fantastic web-based platform and start getting quantitative. And part 4, we try to bring this all together and try to make sense of the data set, the approaches used to produce this data set, and how we can use high content microscopy to generate models to predict organelle shapes and sizes. And we connect this back to the scenario with Dr. G, this antibiotic compound that may have unintended effects on eukaryotic cells, and whether endoplasmic reticulum shape does vary within and between cells. So part 1, what happened to these cells? we focus on the endoplasmic reticulum structure and send students to the visual guide to human cells that is part of the Allen Institute, a multitude of educational resources on their website. And we do this by engaging them in this case study scenario, and the main character, Dr. G, asks his graduate students, well, go visit the visual guide to the human cell, click on their information about the endoplasmic reticulum, and learn about its shape. Next, students compare what they learned from the Allen Cell Explorer Visual Guide to Human Cells, compare this representation of the ER to what they've seen in a two-dimensional textbook or learned in previous courses. And they also learn about the methods to generate the images that you can see on the screen and on the Visual Guide to Human Cells. The next part of this section, we have participants navigate to the Allen Cell Feature Explorer, select Sec61 beta under the protein tags used to visualize cells. And then we have them explore different images as well as plots that are generated on the fly on this web-based platform. And as an example, sample response from a student is included on the slide. The Sec61 beta protein tag is used to visualize the ER. In different cells, the ER fluorescence surrounds the nucleus, but the pattern does vary. So we have students start to observe and ask questions about what they are observing. Participants then dig deeper and begin to quantify nuclear volumes, for example. And the Cell Feature Explorer allows us to plot cell volume and nuclear volume. And students do this by simply clicking on different options. And then they can click on, for example, a dot on this plot on the right of the cell, and then zoom in and learn more about a single cell and its features. And sample student responses include filling out either individually or in teams volumes for 3 cells they pick from the database, and they quantify the cellular and nuclear volumes, learning that maybe there is some variability between volumes. Part 4 brings this all together, and we really try to emphasize how this data set was produced. Thousands of cells were imaged. Many views of a single cell are present, but how were these cell lines created? They were edited cells to include tags to visualize different organelles. This involved automated cell culture, which is another high throughput technique, incubation and plating and visualizing the tags in different multi-well formats, as well as data acquisition using high throughput microscopy. For example, a spinning disc confocal. This created 3D time series that were then analyzed, modeled, and allowed for larger data sets that could be interpreted to create a stain-free organelle prediction as system that the Allen Institute has published on, and students are really interested to learn more about. So we always end the case study with a reflection and have students ask, what was the most memorable concept or skill you learned from this case study? And think about this question and respond in writing. And when we are in person or in discussion forums, they share their responses if they want. The second prompt is, what are you left wondering about? And here the only unacceptable answer is nothing. And how do you start the process of learning the answer? And many times students share that they would dig deeper into the methods used by the Allen Institute for Cell Science and start learning more about some of the publications. And what we really love about this case study is that during the pandemic, it was really adaptable. We were able to adapt this activity to an online format. So beyond the endoplasmic reticulum and this scenario that we created for this case study, this is a rich data set. And students are often really interested in the label-free prediction system that was created based on these high content microscopy data sets. Students often want to continue exploring the data sets and other tools available on the Allen Institute for Cell Science website. And we really start conversations about open data sets, and their responsibilities with publishing open data sets, and how these resources can be leveraged to spark curiosity and emphasize real-world applications of technologies that we may not be able to bring into the classroom or lab, as well as that there are many different perspectives and many different people and disciplines work together to create the data set, to create the cell lines, and to create the label-free prediction system. So with that, I would like to acknowledge our great collaboration with the Allen Institute, our work with Sabrina Robertson and the High Throughput Discovery Science and Inquiry-Based Case Studies for Today's Students HITS Network from the NSF and funding from the NIH. Thank you. And thank you, Kaitlyn.
All right, thank you, Carlos. So with that, we'll invite you to ask questions of our panel. You can leave a comment here on YouTube, or you can email us at education@alleninstitute.org, and we'll pass those on and post responses. So please stay in touch with us here at the education program at the Allen Institute. You can find all of our resources for educators at alleninstitute.org/learn, and you can get in touch with us again at education@alleninstitute.org. And thank you so much.